Hello, and welcome to this Biopharma Dealmakers webcast on regenerative medicine and curative biologics. I am Gaspar Oldenborg, and I will be your moderator today. Today, I have the great pleasure of welcoming 4 speakers who will each explore some of the challenges and opportunities of cutting-edge technologies in regenerative medicine and curative biologics. Our first speaker will be Dr. Jeske Smink, Head of Scientific Affairs at Codon, who will be sharing with us the latest results obtained with her company's autologous chondrocyte implantation-based cell therapy platform for cartilage repair. Our second speaker will be Dr. Jane Lepkowski, President of R&D and Chief Scientific Officer of SIWA Therapeutics, who will be providing us with an overview of Asteria's pluripotent stem cell and allogeneic immunotherapy platforms and their application to spinal cord injury. Third will be Dr. Louis Gruber, CEO and Chief Scientific Officer of SIWA Corporation, who will tell us about SIWA's monoclonal antibody-based therapy to eliminate senescent cells and thus counter the effects of aging. And finally, Dr. Sharon Collins-Persnell, CTO at Organovo, who will introduce us to the high-tech world of 3D printing of human tissues that forms the basis of Organovo's platform for predictive modeling, drug discovery, and transplantation.
I am Louis Gruber, the CSO and CEO of SIWA Corporation, talking to you today about SIWA's technologies for increasing health span by promoting regeneration through senescent cell clearance. At SIWA, we have recognized that throughout life there's an overwhelming process of cellular replication. We each started life as one cell that divided over and over to produce an adult with up to an estimated 70 trillion cells, and 50 to 70 billion cells are replaced each day. There is, therefore, a great reservoir of regeneration in this process. Nevertheless, there's a countervailing process that prevents production of cells from resulting in overgrowth, a process called senescence. You can see in this slide the cell cycle that each cell must pass through proceeding toward replication. When a cell is damaged or stressed, it is stopped from dividing by certain proteins. And an important one is produced by the p16-INK4A gene. This one has been identified as a useful marker for senescent cells and has been successfully used in a number of experiments identifying senescent cells. SIWA's technologies act upon senescent cells regardless of the protein that stops the cell from dividing. We did want to answer why stem cells don't replace aged or damaged cells as we get older, as they did in our youth. The answer we have determined is that senescent cells interfere with other cells, such as adult stem cells, by secreting substances that can cause the other cells to become senescent themselves. Once a cell becomes senescent, it's a dysfunctional cell that's arrested by the p16-INK4A gene product in the G1 phase of the cell cycle. It is prevented from dividing ever again, and this sort of senescent cell accumulates with aging. Over the lifespan of an organism, any cell that has the ability to divide can become senescent as a result of any of a wide variety of stresses, from oxidative stresses to repeated cell division, and even medical treatments such as chemotherapy. Accumulated senescent cells are obstacles to increased health span. The slide you see has a partial list of aging-related diseases which have been connected to senescent cells, as having senescent cells be causal or contributory to the diseases. They range from atherosclerosis to sarcopenia and type 2 diabetes. The solution to this problem is our technology for removing senescent cells, removing the obstacles to increased health span. SIWA has identified a naturally occurring marker on the surface of senescent cells, which we have used to identify an antibody and developed a monoclonal antibody which will bind to the senescent cell marker, causing the immune system to attack, destroy, and remove the targeted senescent cell. And we've demonstrated this all in an animal model. The model for our senescent cell obstacle is sarcopenia. Sarcopenia is a condition of age-related loss of muscle mass and strength that has, in and of itself, been estimated to have a direct health care cost of $18.5 billion in the year 2000, with 45% of older Americans estimated to be sarcopenic. Even so, and despite the fact that the CDC has named sarcopenia as one of the top 5 major health risks, there are currently no therapies approved for sarcopenia. At SIWA, we used a mouse version of our monoclonal Niavira antibody to dose naturally old mice over 20 months of age, twice a week for 3 weeks, with 5 micrograms per gram of our antibody. The treated mice had a statistically significant increase of muscle mass, and that increase was retained in a 9-week treatment-free period after the initial 3 weeks. That persistent result was achieved with no adverse effects, and again, it did not regress even though treatment was stopped after 3 weeks. Tying our results to senescence, we also determined that untreated old mice had about 3 times as much expression of P16-INK4A gene product as did untreated young mice. And when treated with our Niavira antibody in mouse form, it was observed that the P16-INK4A level went down to that approximately of untreated young mice. At SIWA, we have technology for clearing senescent cells that includes Niavira antibodies in mouse and human form, and also other technologies we've developed for clearing senescent cells. Because we have identified a cell surface marker for senescent cells, we also have the potential of developing vaccines against senescence. We see that senescent cell clearance, as shown by our results and results of other laboratories around the world who have examined senescence, that senescent cell clearance provides a comprehensive therapeutic for age-related diseases, such as sarcopenia and complications of type 2 diabetes. We're pleased that with our antibody, lasting results were obtained through twice-daily injections, not requiring infusion. And we also employ a natural process for removing deleterious cells. As the body removes potential cancer cells and removes other types of deleterious cells with the immune system, the antibody-dependent cellular cytotoxicity is a process that we make use of as well. And so it fits well with the natural operation of the bodily processes. Also, by using our antibody to clear senescent cells, obstacles to stem cell therapies can be removed. Recipients can be made less resistant to the stem cell therapy, and donors can have potentially problematic senescent cells removed from the transplant. Of course, being an antibody therapy, we fit in well with the well-established strengths of the antibody therapies that are currently available for numerous diseases from arthritis to cancer. We have, over the years, had patents granted, allowed, or issued on all or part of our technology around the world, from Australia through China, in the United States, in South Africa, to Europe, and numerous other countries as well.
Thanks, Louis.